Good evening, Raiders, and happy Wednesday to you. This is Coach Buse, and I'm going to be sharing some slides with you on classical genetics this evening. I'm going to go ahead and present my screen right now so that we can take a look at some of these together. Give me 1 second. Sorry about the background noise. I'm upstairs, and I'm going to go ahead and pull up the presentation on Mendelian experiments with probability. And so you can see some pictures of some different of some birds here, and you can see some of the variation in colors and plumage on the birds. And of course, we know that that's from meiosis and sexual reproduction and some of the concepts that we talked about prior. And so let's take a look at Gregor Mendel. And so Gregor Mendel is the father of genetics. He developed the laws of heredity. He worked with pea plants, and he was extremely lucky because the pea plants worked in very specific ways. And so, for example, out of the traits that he observed, the 7 traits that he observed, there were 2 distinct classes for each of the 7 traits. For example, purple flowers and white flowers, tall and short plants, axial and terminal buds, and Gregor Mendel was an Austrian monk. He worked he worked in a very quiet place where he was able to collect some very important data. Unfortunately, his data was not discovered until after his death, and he is celebrated as the father of genetics. During this presentation, I wanted to take a minute to review with you some of the structures of plants and some of the skills of plants. And so this slide talks about the basic body plan of land plants, the roots versus the shoots. Recall that plants grow from their meristem, which is the tip of the roots and the tip of the shoots. Make sure that you understand the difference between determinate and indeterminate growth. An example of determinate growth would be a plant that stops growing at a certain point, versus indeterminate growth would be like grasses, which just continue to grow throughout their life cycle. Primary and secondary growth in plants, number 3 there. Primary growth is the growth up towards the sky and down into the ground, whereas secondary growth is growth that grows wider girth. The ploidy of different stages in the life cycle of plants. Most land plants spend a portion of their life cycle as a haploid plant and a portion of their cycle as a diploid plant. This is an example of a diploid organism. It has 2 sets of chromosomes. The seeds inside the fruit are also diploid. The pollen that is produced by this plant would be an example of a haploid cell. Double fertilization in angiosperms, so flowering plants have a double fertilization inside the seed. The first fertilization is for the embryo, and the second fertilization is for the endosperm, which provides nutrients for the developing embryo. Number 6 down here says explain the differences between the reproductive structures of plants. Just be aware that there are different ways that plants reproduce. Some plants reproduce using spores. This would be mosses and ferns, whereas flowering plants reproduce using seeds. Seeds are a much more effective way of reproduction because they can withstand changes in the environment more easily. Vascular plants, the vast majority of plants that we see are vascular plants. They have a very specific shoot system, which is the above ground system, and roots, which is the below ground system. The root systems can be either a tap root, which is shown here, or a fibrous root, which you could see if you looked at the roots of corn or grass. They have more of a fibrous root system. The meristems of plants allow the plants to grow up and to grow wide. The apical meristems allow plants to grow taller, and so this would be an example of the shoot apical meristem and of the root apical meristem. We took a look at those during our unit on mitosis. You looked at some of those cells. The lateral meristem allows plants to grow wide and thick. Not all plants have lateral meristems. Woody plants have very extensive lateral meristems, which allow them to grow wider. An example of those lateral meristems would be the growth rings that you would see in some species of trees. Leaves, recall from our section that leaves are the photosynthetic units of plants. They allow plants to absorb light. They allow plants to exchange gases, and in many cases, they have a cuticle to prevent water loss. This is the primary organ of photosynthesis in plants. Indeterminate versus determinate growth, I mentioned that a little bit earlier. Indeterminate growth would be growth that doesn't stop versus determinate growth, which stops usually at a certain point. And so which type of tomato plant would a gardener prefer between the two? Of course, indeterminate growth would be better for a gardener because the plant would continue to grow over long periods of time and would not be stopped at a particular moment. Plant reproduction. Plant reproduction falls into two different categories. Monoecious plants have male and female flowers on the same plant. So in a moment, we're going to be talking about pea plants and Mendel's experiments, and that would be a monoecious plant. Other plants, for example, papaya, the cannabis plant, are dioecious, which means that they have separate male and separate female plants. Another example that's not on the slide here of a dioecious plant would be a ginkgo tree. Ginkgos have both male and female trees. Male gametophytes are the pollen grains. Pollen grains are formed by meiosis. This shows some pollen, some different structures of pollen. Pollen contains the male gamete, which is formed through meiosis. And these pollen grains, of course, are what cause untold allergies during the spring season, especially in areas where there are a lot of flowering plants. So if you're suffering from allergies, it may be because of some of these pollen grains. The female gametophyte is found in the ovary of the plant. In the female gametophyte, only one of the 4 megaspores that's produced by meiosis survives to become the egg cell. And so here you can see the egg cell. This is the egg cell that will be fertilized to make a new embryo. And gymnosperms and angiosperms, these are different species of plants. Gymnosperms are the cone-bearing plants. Angiosperms are the flowering plants. And so again, in this first little section, just providing you with some background, when fertilization occurs, there are 2 sperm cells that fertilize the egg cell, that fertilize the 2 parts of the ovary, excuse me. The first sperm cell is going to fertilize the egg. The second sperm cell is going to fertilize the polar nuclei, which is going to form the endosperm. The endosperm will provide the food for the growing embryo. The ovary will then develop into a fruit. How do we know if something is a fruit or a vegetable? Of course, in biology, a fruit refers to any seed-bearing structure. Anything that produces a seed is a fruit. So strawberries are a fruit. Tomatoes are a fruit. A pea pod, which Mendel used to study, is a fruit. Peas are seeds. They will grow into a mature adult pea plant. Meiosis and fertilization ensures that each one of these peas is unique from one another. And so we're going to be looking at some experiments that are similar to Mendel's. Here, we're going to be using, I was hoping to use Wisconsin Fast Plants in the class. We're going to be using our corn genetics to show the laws of heredity. Monocot versus dicot flowering plants. We looked at an example of a dicot plant during semester 1 with our marigolds. A dicot plant has fibrous roots. It has net-like veins and flowers in multiples of 4 and 5. So again, so far, let's not stress too much about plant biology, but I would like for you guys to be aware of that because it's something that you didn't really study in your introductory biology class and that you should be aware of. So let's get back to Mendel. Mendel studied 7 traits and he made control crosses between 7 contrasting traits. He crossed a true breeding round versus a true breeding wrinkled, a true breeding yellow versus a true breeding green, and a true breeding purple versus a true breeding white, and so on. And one important thing to understand about pea plants is that there's 2 ways that they can reproduce. They can reproduce through self-pollination, which means the male and female part of the same plant are involved in reproduction, and cross-pollination, which means that the male and female plant of 2 separate parts are involved in pollination. So to avoid self-pollination, Mendel removed the anthers from the plants. And by removing those anthers, he was able to prevent the pollen from one plant from self-fertilizing, and this allowed him to make the controlled crosses. Mendel started with true breeding plants. These plants are what we call homozygous. Homozygous, excuse me. Homozygous means the same. So for example, when Mendel crossed his true breeding purple and true breeding white, what was expected at that time was that he would end up with a blended inheritance. He would end up with something that was in between. And that's not what he observed. He observed that for each of his 7 traits, they exhibited just one class. So when he crossed purple and white, he ended up with purple. And based on this observation, he developed the principle of dominance. And so the principle of dominance states that in a particular cross, one trait will be dominant and the other will be recessive. So in this particular cross, which of the traits is recessive between the purple and the white? Okay, so if you said that white was the recessive, you are 100% correct. And how do we know that? White is not shown in the first generation. Now, what Mendel observed is when he self-fertilized these first generation plants, he ended up seeing about 25% of them show that white trait in the next generation. And so this is how Mendel developed what was called the principle of segregation. The idea being that this flowering plant, even though it was not purple, or sorry, even though it was not white, carried the genes for the white flower color. And so today we use terms like dominant recessive. We use letters to represent the genotypes of the plant. For example, big P is purple, big P is purple, little p is white. And so when we perform some crosses, this gives you a chance to do some practice here. Excuse me. Let me back up here. So when we do crosses using Punnett squares, we place the genotypes of the parents on the outside. Now this parent here, who's a true breeding purple, can pass down either a purple allele or a purple allele, and this one can pass down a white allele or a white allele. And what we end up when we cross these is we end up with either a big P, big P, purple plant. So it's not possible to do a cross between these organisms and get a white plant. And if we take a look at this cross here, this explains Mendel's F2. When we do a cross like this, all of the offspring would end up purple except for the one in the bottom here, which would be white. So Mendel would expect that about 25% of his plants would be white based on this cross, which is consistent with his data. So take a minute if you could and practice these. I'll give you a second if you could pause the video and do these crosses, and I'll check in with you in about 2 minutes. Just pause the video real quick. Okay, so if you did the first cross, you should see that 100% of the offspring would be expected to be round, versus 0% that would be expected to be wrinkled. That would be our phenotype or our physical appearance. 100% of them would also have the genotype big R, little r, based upon that square. In the second cross, when we do our heterozygous cross, we would expect for the phenotype, about 3/4 of them would be um, round, and about 3/4 of them, sorry, excuse me, 1/4 of them would be wrinkled, based upon these parents. The genotype would be 1 to 2 to 1. So about 1/4 would be big R, about 1/2 would be big R, little r, and 1/4 would be little r, little r. So to review some terms with you really quickly, take a minute to review these. F1 versus F2, generation, true breeding versus hybrid. Remember, a hybrid contains one gene that's dominant and one that's recessive. Self-pollination versus cross-pollination. Homozygous versus heterozygous, genotype and phenotype. And then gene versus allele. Now, I haven't used the term allele with you guys yet, but an allele is simply just a version of the gene. So an allele for purple flowers would be represented as big P, and allele for white flowers would be represented as little p. So the connection between meiosis and genetics. So I started you guys off with a little discussion on meiosis, and now we've gotten into genetics. So the reason that Mendelian genetics works is because of the separation of homologous chromosomes. So here you can see the homologous chromosomes represented in red and blue, and these homologous chromosomes are separating into distinct cells. And because they separate into distinct cells in that way, this makes our Punnett squares work. Dihybrid crosses. So these are a little bit more challenging. And so a dihybrid cross represents all the possible gametes that each parent can contribute. So for example, if a parent has the genotype big R, big Y, little y, what are the combinations that would be possible when we set up our Punnett square? Well, to set up our Punnett square, we're going to have only one of these two letters be passed down to the offspring. So for example, we could have a big R, big Y, a big R, small y, a little r, big Y, or a little r, little y. So there are 4 different combinations of gametes for this particular genotype. And so I'm going to give you guys an opportunity to set up this square. And so if you could pause this real quick, and then I'll go over how to solve this. And go ahead and do that. Okay, so when you set this up, you're going to set up the parents on the outside, and you're going to set it up the parents as big R, big Y, big R, little y, little r, big Y, and little r, little y. And you'll do the same on the left side. In the inner boxes, you're going to combine like letters. I'll repeat that one more time. You're going to combine like letters. So when you combine the like letters, you will get the genotype of the offspring. Now, when you complete the box, if you've done it correctly, and we'll work through a couple examples over the next couple days, you'll see a combination that looks like this: 9 to 3 to 3 to 1. This is something that you can just memorize. When you do a dihybrid cross using Mendel, you're going to end up with a 9 to 3 to 3 to 1 ratio. And you'll see a similar ratio in the corn genetics experiment that you're working on later this week. So consider the cross between the plant with round seeds, purple flowers, to one with wrinkled seeds and white flowers. So when we do this experiment, if you want to do this faster, what you can do is set up 2 small Punnett squares. So you can set up the big R, little r, and the little r in one square, and you can set up the big P, little p, and the little p in the second square. Once you have both squares set up, you can simply just multiply the two squares together to get the total probability. For example, if you wanted to know how many round seeds and how many purple flowers you would get out of this cross, you could run two small squares. So for the first cross, I would know that one half of the offspring could be expected to have round seeds. I would also know that one half of the offspring would be expected to have purple flowers. One half times one half equals 1 over 4. So my conclusion would be that 1 over 4 of the flowers would have round seeds and purple flowers. Take a moment to review Mendel's laws that we discussed. Mendel's laws help to explain why these dihybrid crosses work. Segregation, dominant recessive, we've discussed. Independent assortment. Notice how each trait is on a different chromosome. So A is on this chromosome and B is on this chromosome. And because they're on separate chromosomes, they separate independently of each other, which allows it to follow the Mendelian pattern. So again, with probability, it doesn't matter how often you flip a coin or how many times it's already shown heads, the probability is always 50%. So if I toss a coin 4 times and it comes up heads every single time, the next time I toss it, the probability is still going to be 50%, right? And so this is how Las Vegas was built. It's based upon this idea that people think that they're having a losing streak or that they're having a winning streak. It's all probability. There's no such thing. Genes come in pairs, chromosomes come in pairs. Genes are located on chromosomes. And so when we go to the next step, we're going to talk about how we can use our knowledge of Mendel and move it into the next level. And so we'll talk a little bit more about that chromosome theory next week. Keep practicing with your process. Hold on just a second. Get yourself comfortable with working through some of those crosses. You guys can ask me questions. You can work through some of the different sample problems that are available out online. Take a look at some of the resources that are out there for students. This week, I really want you to get a mastery over the single factor crosses and the two factor crosses. So, so take the time to practice through those, and we'll talk a little bit more tomorrow afternoon. Thank you guys for your attention, and I will see you tomorrow. Good night.